So I have talked about microplastics on many occasions, not only on my podcast, but also in some of the public discussions. Just recently, it happened in Mangalore, India. It was a lit fest, but anyway. So you can find that article in the newspapers right now. And obviously, I got alarmed by this news article from The Guardian, which is an excellent newspaper. Uh, a bombshell, doubt cast on discovery of microplastics throughout human body. That article talks to many scientists, refers to a few articles, especially the one that reported increasing amounts of microplastics in human brains, and falls to some, points to some problems with detecting plastic because apparently fat in the body, especially brain, is made up of so much fat that can confuse the detection of plastics, which means you get false alarms. And nanoplastics can cross over into bloodstreams, but they are very hard to measure and detect, and so on and so forth. So the article points to a letter written by a number of experts in this area in Nature Medicine that came under Matters Arising, a letter that says challenges in studying microplastics in human brain. I'll read some text from here because this is an important topic and another example of how science can often get excited about things and go a little bit overboard. And as an outsider, I want to worry about microplastics, especially in the oceans and everywhere else in the air, in the food, etc. I also have always said the causal links to health outcomes are not always obvious, but nonetheless, as an outsider, I cannot be very sure of just how reliable are the papers, even when they are published in reputable journals. So these articles help. Human exposure to microplastics and nanoplastics (MNPs) is an emerging concern, and many papers claiming that they are accumulating in human tissues. In a recent study, Nihart et al. Used the analytical technique of pyrolysis gas chromatography mass spectrometry to detect MNPs in human liver, kidney, and brain, reporting the highest concentrations in the brain, with polyethylene as the predominant polymer. This study, as reported, appears to face methodological challenges such as limited contamination controls and lack of validation steps, which may affect the reliability of the reported concentrations. In this Matters Arising, we highlight methodological limitations that have general relevance for advancing robust and reproducible MNP detection in human biomonitoring studies. So, this method of pyrolysis gas chromatography mass spectrometry basically says the best way to detect plastic amounts is to vaporize it and then capture the vapor and then measure it. This is what this is referring to. Lack of procedural controls and quality assurance measures are one of the flags raised. Detecting MNPs in human tissue poses important analytical challenges due to the widespread presence of plastic contaminants and the complexity of biological matrices. International best practices guidelines such as emphasize the need for comprehensive and consistent quality assurance and quality control procedures. These include the systematic use of procedural blanks, field blanks, matrix contamination assessments, etc., which are, technical terms that are beyond me, but we should be aware of these. So these measures are essential to differentiate true tissue-derived MNPs from contamination induced during sampling, sample preparation, and detection. So the Nihart et al. Study basically had issues in the quality assurance, quality control, and many other details like the contamination from reagents, labware, and airborne particulates, which may have influenced their findings. Moreover, the tissue samples in the study were collected across different years and locations, raising uncertainty about whether harmonized contamination minimizing protocols were consistently applied. So let's assume that that study is basically highly doubtful. Nihat et al. Also used a 10% potassium hydroxide digestion at 40 degrees C for 72 hours, followed by uh, centrifugation and ethanol washing to remove tissue matrix. So more details, technical details as to why the results are doubtful. However, potassium hydroxide digestion is known to be suboptimal for lipid-rich tissues such as the brain, with incomplete tissue removal often resulting in residual particulates that can be misidentified as synthetic polymers. Basically, fat apparently can be mistaken for polyethylene, and brain is 60% fat. So, if you are dealing with trying to find microplastics in the brain, then the chances of false detection are very high. Okay, that's kind of the summary in words. Uh, the other details I'll skip. The study does not report polymer spiking experiments to validate recovery efficiency, limits of detection and quantification, or identification accuracy, nor does it describe the approach used to assess the possible impacts of the extraction method on particle integrity. So, there are many technical details. They have to provide this because if they are objecting to published paper, then they better say exactly why they are objecting. So they are providing all the technical details. Okay, so we'll skip that. But here, limitations of the pyrolysis and gasification and mass spectrometry approach for micro and nanoplastic detection in tissues. PYGCMS is a well-established tool for identifying and quantifying pure polymers, but its specificity is limited when applied to complex biological samples. The consistent predominance of polyethylene across all organs, time points, and individuals in the study by Nihart et al. Is notable, as environmental exposure studies often demonstrate greater diversity in polymer types. So not all microplastics are the same. So this detection method is sensitive to that. Many biological lipids, especially long-chain fatty acids, produce pyrolysis products that mimic polyethylene mass fragments. So that's where the false alarms can come from. Jumping again ahead, distinctive signal characteristics of specific polymers and verifying consistent retention patterns was a problem as well. These patterns reflect the expected positions of polymer peaks relative to adjacent compounds, such as alkanes, in the chromatogram, which help differentiate polymers from similarly fragmented biological compounds. So the authors noted that such marker ions were present, raising concern that the observed polyethylene signals may have come from residual fatty acids rather than synthetic polyethylene. So instead of the presence of microplastics, they must have just, they might have just taken fatty acids as polyethylene. This possibility is reinforced by the lipid-rich nature of the brain tissue, 60% lipid by dry weight, so high fat, which means high false detection possibilities. All these things together, then the detection of MNPs in human tissues continues to face substantial methodological challenges compounded by the lack of harmonized reporting standards across the field and their limited implementation in scientific journals. Studies quantifying MNPs in biological matrices should follow the rigorous quality assurance quality control protocols, including clear false positive identification strategies, consistent use of sampling and processing blanks, contamination controlled procedures, and validated digestion and recovery methods using well-characterized reference materials. Given the complexity of human tissue matrices, the use of PYGCMS requires particular caution. In the absence of more targeted methods, accurate polymer identification should rely on multiple analytical approaches to ensure specificity alongside characterization of size and shape. The challenges reportedly by Nihart et al. Illustrate broader ongoing gaps in analytical rigor within the field. As human biomonitoring of MNP advances, it's essential for the scientific community to adopt harmonized practices that promote transparency, reproducibility, and confidence in reported findings. Notably, findings that lack adequate validation may inadvertently shape public perception and regulatory discussions despite underlying scientific uncertainties. Ensuring methodological robustness is, therefore, critical for producing reliable data to support risk assessments, inform policy decisions, and guide future research. Plastic is a menace. It's all over in the oceans, in the air, in foods, and in our bodies, maybe. But just how extensive is it? Has it crossed into bloodstreams to reach across the blood brain membrane into the brain? If so, are there causal links to cognitive abilities and Alzheimer's, or in the testes and in fetuses, in the womb, and so on and so forth? So serious scientific questions, but in the meantime, we have to be careful. Okay, scientific methodologies have to get much more rigorous, which is the main point of this letter in matters arising in nature medicine. Okay, I have a short on this where I'm going to link this podcast just to give a short, brief message to the public that we all have to be careful about this. Okay.